Thank you so much, Carmen. Uh, Good morning, everybody. I realize that this panel is standing in between you and lunch, but we have really exciting panel today that'll be talking about natural history studies and the vital role they play in advancing research and treatments for rare disease. So today we have a really exceptional group of experts and advocates who'll be sharing their experiences, challenges, and successes, and of course, in developing and sustaining natural history studies for rare disease communities like ours. So starting with Kasha, to my far left here, could you briefly introduce yourself to the audience, telling us your name, your organization, and role there, and we'll kind of go down the line.
Good morning, everybody. My name is Kasha Morris, and I'm a co-founder of the Tango 2 Research Foundation and mom to Ryan, who has Tango 2 deficiency disorder.
I'm Liz Cassidy. Is my mic on? I'm Liz Cassidy. I'm the Senior Manager of Research Projects at the TSC Alliance. I kind of did my toes in several research projects at the TSC Alliance, but I mostly help manage and oversee our TSC Natural History Database and Biosample Repository, which I'll be talking about today.
Hello, I'm Browning-McNee. I am with the Reagan-Udall Foundation for the FDA. We are an organization that is a 501c3, so we are not part of the FDA. We are not a government agency, but we are a 501c3.
Awesome. Thank you, ladies. And again, my name is Deena Chisholm. I'm the Director of Research Engagement at the Tango 2 Research Foundation. But before we jump right into getting some information from these ladies, I want to know what you guys think. I want to get a sense of, with this first polling question, your thoughts around natural history studies. Are you going to load the polling question? All right, while he's getting that loaded, so natural history studies have numerous benefits. So what we're interested in finding out from you all today is, what do you believe is the most significant when it comes to rare diseases? Is it advancing scientific understanding, improving patient care, identifying new therapeutic targets, or facilitating early diagnosis? All right, what do we think? All right, it seems like the answer is D. So everything, right? All of the above are really important when we think about rare disease and natural history studies. So with that, let's dive right into the discussion. Let's first start with Kasha. So Kasha, you've been at the forefront of the Tango 2 Research Foundation advocating for patients with Tango 2 deficiency disorder. Let's kick things off. Can you tell us a little bit about Tango 2 deficiency disorder and your connection to this rare disease?
Okay, well, Tango 2 is a gene on chromosome 22, and it's an acronym that stands for Transport and Golgi Organization. And to be honest, we still don't really know the function of Tango 2 in a healthy person. We're still working on that with research, but we do know that it's characterized by progressive developmental delay, intellectual disability, movement disorders such as ataxia, dystonia. Many have seizures, speech delay, and progressively gets worse. And on the worst side, kids with Tango 2 are at risk for metabolic crisis and potentially fatal heart arrhythmias. My connection to Tango 2 goes back to my son. Tango 2 was just discovered in 2016 as a gene related to human disease, and my son was diagnosed in 2017 after 15 years of searching for a diagnosis. And it had newly been discovered. There were only 2 research articles about it, very little information, and the prognosis was poor. So as many of you know, that's devastating. And my husband and I decided that we can't just do nothing. We can't just wait for this disease to take our son. So in 2018, we started the Tango 2 Research Foundation in hopes that we could somehow speed up the process and get more research done on this disease that could help our son. So that's where it starts from.
Yeah. And given that the Tango 2 Research Foundation is relatively new, can you share how the natural history study got started and what progress has been made in just the 6 years since the Tango 2 Foundation was started?
Well, right after we got the diagnosis, one of the first things I think many families do is you try to find other people with the diagnosis. And we found that in a Facebook group. There were 9 other families. And for the first time in 15 years, I felt that connection to others who really got me. And so in that Facebook group, I started to take notes. I just wanted to know everything that these families knew. What symptoms did their children have? What had they tried? What had worked? What hadn't worked? And I started taking these notes and putting them in an Excel spreadsheet. And, you know, the reality is that that kind of was the start of our natural history study, was those notes. And ironically, knowing what we know now, it really did begin a decade or more before the disease was even discovered. The best nugget that has come out of our natural history study goes back to when my son was 2 in 2003. And his quality of life was terrible. He didn't have the strength to hold his head up. He didn't have the strength to open his fingers and hold a toy. And he had lost the skills he had once had. And I remember as a mom, you just feel like it's your job to make things better. You got to fix this. And I remember it clearly because it was Halloween and he was Bob the Builder, and, you know, whose motto is, can we fix it, right? And I gave him a Flintstones vitamin, probably a shot back to my own childhood where that's what we took, Flintstones vitamins, right? So I gave him a vitamin just for the lack of anything else to try, because we had been to dozens of doctors, hundreds of tests. But I gave him this Flintstone vitamin, and the next day I noticed some improvement in some of his symptoms. And the day after that, some improvements. And this day after that, some improvements. And, you know, it would still be 15 years before we got a diagnosis. And throughout that journey, my husband was relentless in bringing up those vitamins every time we saw a new doctor. And, but without a diagnosis, you know, it never seemed to be relevant. It never seemed to be something so simple could possibly explain a disease that so impacted his life, right? But once we had a diagnosis, we took that spreadsheet and gave it to Dr. Christina Miyake, who is a clinician researcher at Baylor College of Medicine, and she works at Texas Children's. I gave her those notes in the Excel sheet, and my husband once again brought up those Flintstone vitamins, and she listened. And there were only 12 families in our natural history study at that time. And she started to ask other families about vitamins. And just 1 year after we had started the foundation, we had a family conference, and it was already, a pattern was forming. Kids like my son, who had never had a metabolic crisis, were taking vitamins. And kids that had metabolic crises and kids that had passed away from Tango 2 were not on any vitamins. And that was kind of the start of it. And since then, it's only become more apparent. We now have animal models that are proving that vitamins, particularly B vitamins, are making a huge difference with our kids. We've got basic lab research that's going on to still try to support that. We've got iPSC cells that are showing B9 corrects arrhythmias in Tango 2 heart cells. So the evidence is only getting stronger, but it started with those 12 families in that Excel spreadsheet.
Yeah, that B vitamin breakthrough story is nothing short of amazing and what everyone kind of hopes for with developing a natural history study. It's literally taking the information and noticing those patterns and finding a trusted clinician researcher who will listen, right? That's so valuable. Kasha, rare diseases, like you mentioned, just that cohort of 12 families, they often have really small populations, which can make establishing a natural history study seem really daunting, right? What would you say to organizations that feel like they don't have the numbers to support the development of a natural history study for their own rare disease?
Well, I think you don't have to wait for a certain number. You know, you don't want to wait. You want to start collecting those patient experience stories and getting them together collected in one space, because like I said, it started with 9, and then by the time we had 12 patients, we were finding a pattern. So you just never know.
Yeah, and today our natural history study has over 100 patients in just 6 short years. So yes, there is powers in numbers. Find those people, engage them, encourage them to join your natural history study. It's really important for seeing those advances in our communities. I know that establishing a natural history study can seem overwhelming at times, right? Are there particular steps or prerequisites that organizations should follow when they develop a natural history study? One, and then what has your experience been in setting up the Tango 2 natural history study? You alluded a little bit that you had an Excel spreadsheet.
Well, we've been at this for 6 years, and I have to tell you that it hasn't been our experience that we did step 1 and then step 2 and then step 3 and then step 4. You know, that, yeah, oh, you have to check the box with these prerequisites before you can go to the next thing. It seemed like a lot of things have been going on at the same time, you know. So we started the natural history study with the Excel spreadsheet. At the same time, we were trying to fund basic science lab research to understand the basic mechanism of Tango 2. We were also trying to get a biorepository up and running at our very first conference with, I think we had 15 affected individuals there, taking some blood, taking some tissue samples, mouse models, animal models. We were trying to get those going. So in our first year, we had a number of initiatives that were going, and they were not linear.
Absolutely. So you were right, as the founder of the Tango 2 Research Foundation, and of course, as a parent of Ryan with Tango 2 deficiency disorder. From an advocacy standpoint, what role has the foundation played in facilitating and sustaining the natural history study for TDD?
I really see the Tango 2 Research Foundation as like the center of a wagon wheel, right? And our role is to be the connector and to foster the collaborations among everyone who cares about this disease. So, you know, for example, we connect families to each other. We connect researchers to each other. We connect clinicians to each other, and then families to clinicians, and so on and so forth. That connector, that link person who links them together. And we also look for those collaborations. You all know, you know, with rare disease, time is your enemy. So if we can help facilitate something being done collaboratively that avoids redundancy or duplication where it's not needed, then we want to do that. So if we hear of a researcher who needs cell lines and we'll connect them with someone who's got them. If we hear of a researcher who needs knockout mouse models for Tango 2, we're going to connect them with someone who's already got that to try to facilitate their work in the most efficient way possible. So we collaborate, we foster the collaboration, and we make those connections.
Yeah, yeah. Looking ahead, what's next for the Tango 2 deficiency disorder natural history study? Can you give us a glimpse of what the future may hold?
Our natural history study has really been a retrospective natural history study to this point. So we do want to try to put some effort into getting establishing a prospective natural history study. So we're putting a proposal together for that and looking for funding avenues to get that work started. And kind of while we look for those avenues of funding, we're trying to find as a foundation, what can we help with? So we might be starting with a piece of it, like what can we do to fund a global health index? That might be a tool used in a prospective natural history study. Let's start with that so that we're not just waiting for the funding and the project sitting there. We're trying to see what can we start to chip away with. So that's what our next endeavor is heading towards.
Absolutely. So again, thank you, Kasha, so much for those really valuable insights and sharing, and such a powerful story about your journey with TDD and the foundation's efforts. So audience, get those phones out again. Transition to our next polling question, shift in focus for a minute here. We now know biorepositories play a really important role in many of those natural history history studies, but the level of familiarity with them can vary. So I want to know from you all, how familiar are you with the role of biorepositories in these studies? Are you very familiar, somewhat familiar, or is this a newer concept for you? All right, let's see where we landed. Okay, some familiarity. Okay, some new. Okay. Uh, Liz, can you tell us a little bit about the role of the TSC biorepository and the TSC natural history study, how that got started, and what the experience has been so far?
Yeah, I love that you asked the biosample repository question before my section, because I love talking about it. Just for anyone that's unfamiliar, the TSC Alliance, we started our natural history database back in 2006 from a federally funded grant with a collaboration with a researcher and clinician in Texas. So we started collecting clinical data back then, and now we've grown kind of a lot since then, obviously, and we now have over 20 clinics participating in the US and 2 in Canada on our natural history study. On top of that, we are also able to remotely consent and enter data through the TSC Alliance. So myself and a colleague are able to obtain medical records from clinics that may not be participating in our project. So we're able to do data entry into our platform study tracks there as well. And in 2016, we actually started to collect biosamples that are associated with the patients that are enrolled in our natural history database. So now those IDs for the NHD enrollees are also associated with these biosamples. So our biosamples are things that we've collect from the human specimen. So we collect annual blood samples. We can collect things like DNA from cheek swabs that actually are preferable for our little kids that maybe not want to get stuck, or someone that's needle phobic, giving them the options for them to participate in that. We also have the opportunity for them to coordinate the donation of tissue samples as well, which is really helpful for things that may be looking at tissue in affected tumors from organs in TSC. So things like those kidney tissues, brain tissues, things like that. And we try and coordinate with either historical tissue that may be stored in pathology in these clinics, or with an upcoming surgery for these patients as well, trying to see whatever's not used in pathology for clinical purpose, if they're able to donate that to our biosample. So we've now, since inception, now the two protocols have merged. We've collected over 2,700 biosamples. The large majority of that is those blood samples that we collect annually. And we can't do that without those clinics that you see in those push pins and gray buttons over on that map.
Wow, 20 active natural history study databases. That's incredible. You mentioned some exciting projects that stem from the TSC Natural History Study, including research in reproductive, perinatal health, and some neuropsychiatric disorders. Can you elaborate a little bit on these projects and how they've kind of contributed to TSC research?
Yeah, absolutely. So you'll see on this slide just a little bit that I mentioned that we started in 2006 and we've really grown. And I won't go through all of those points, but that we have learned from those experiences from decades of doing this project and where we need improvement. And one of the things that I always like to tell people and highlight is that we make these improvements through the patient voice. One of the things that we try to do every 5 years is have a constituent survey where we try to collect information of what our community wants our organization to focus on. We also hold a world conference with our TSC community every 4 years. And our last world conference was held in 2022. And in that world conference, we had a session with community members where they told us that one of the things that they wanted to see more data and more conversations with their clinicians was on women's health. How can they start having conversations about getting pregnant, concerns about passing TSC along to their children in pregnancy, and due to the genetics of TSC being autosomal dominant. So one of the things was that we saw is there's such a lack of data on women's health and pregnancies, and that we really wanted to gather more data. And one of the gaps that we found is that we weren't collecting this data longitudinally in our natural history database. So we decided that that was definitely something we needed to add. So that's just a little bit of an example of data that we have just started collecting this year. So we're continually making improvements. Even though we have started for so long, it's always a moving process. And I think that we're always looking out for what our community wants us to focus on and how we can later use that on for data points that may be useful in things like preclinical models or clinical trials that we may give data and biosamples alongside for our researchers.
Yeah, TSC seems like they have a very vested and engaged stakeholder group. Given that the TSC Natural History Study, like you said, has been running for many years now, over a decade, it's bound to have encountered some challenges along the way, right? Can you talk a little bit about some of those roadblocks that TSC may have faced and how you managed to overcome them in your organization?
Yeah, I think some of those roadblocks that we've faced have, of course, was COVID. We had a little bit of a hit of a pause of data that we were collecting, gaps in data from patients maybe not going to their clinics, from medical records that weren't being updated, things like that. Of course, we have to look out for the types of people that we are having to, the clinics that we are also going to be funding for this project. And that's, of course, something that we always try to reach out for our investors and sponsorship to be able to fund this project. I think some of the initiatives that we try to take is really to show that how much we've grown and getting people to participate and to really emphasize the impact. So one of the things that I'm actually going to show on the next slide is that community impact is really important in funding this project, and we really can't do it without them. So this is just kind of a little bit of that example that the community impact from their participation has really helped us grow projects like quality improvement projects that kind of stem from that, reproductive and perinatal health task force project that I talked about. It kind of branches down into the impact into our researchers and academics that we also give to for this project, as well as with our industry partners. So this is just an example of some of the biosamples and data that we've contributed for clinical trials. We have an ongoing clinical trial looking at ganaxolone where they used plasma samples from our biosample repository. That's ongoing phase 2 clinical trial. And that trial is currently underway. And obviously we have our researchers, academics that are using our biosamples and data as well. We have over 20 publications now from biosamples and researchers. And of course, this is growing. Researchers continuously try and request our data and our biosamples for use through the use of a steering committee where they go through an application process, making sure that it's being impactful for our community is the important part, and that they, of course, are HIPAA compliant, all of those things that our IRB looks out for, and that they remain collaborative with us at the TSC Alliance.
Yeah, you mentioned something really important, that you guys are looking to expand. When you think about expanding globally, can you speak a little bit to the importance of, that expansion and kind of what it means for TSC research moving forward?
Yeah, that's one of the things that I'm really passionate about is, as you saw on that map earlier, that a lot of our sites and clinics are US based. We have now started to grow into Canada with 2 sites in Montreal. But we are really starting to focus on more of a global representation of TSC in our natural history database. And we are now in collaboration with the TSC Alliance Mexico to hopefully start the natural history database there and to hopefully expand in more Latin American countries. As someone that is Latina herself, I think that that's very exciting to think about. And I hope that that paves a pathway for other rare diseases that may have natural history studies that are looking to expand. And so we're looking to kind of use that as our pilot for a global initiative in the natural history study and hopefully expand to other countries such as Europe, and collaborating with ongoing TSC associations that are international. So we have a TSCI consortium, which really stands for TSC International, and they are helping us collaborate with ongoing research initiatives and looking to continue to have continue discussions with them on how we can have research going on in their countries.
Yeah, awesome. Thank you, Liz, for sharing TSC's successes, challenges, and next steps. TSC's growth is both inspirational and incredible. So before we talk about the broader role of natural history studies and patient registries, one thing that I think all of us can kind of, agree with is that recruiting participants for a natural history study can come with its own set of challenges. So last polling question, what do you think is the most critical issue to address when it comes to recruitment? Ensuring informed consent and reaching a diverse population, securing long-term funding, or maintaining engagement over time, or maybe all of these challenges are equally important. All right, let's take a look at those results. Okay. All right, all of these. Okay. It seems like a general consensus. So this time we'll turn it over to Leanne. What role does natural history studies and patient registries play in developing treatments for rare diseases?
Thank you, Deena. And I have to say, I'm not remotely surprised by the answer to that last poll question. All of these challenges are critical because, quite frankly, the outcomes are so critical. So, when we're thinking about natural history studies, patient registries, it's important that we really think about that as real world evidence that we're collecting, and real world data that we're collecting, real world evidence that we're building. And, I think one of the big challenges is also thinking about how do we ensure that the work that you're doing, the time and effort that you're putting in, is also fit for regulatory purpose. And so we'll talk a little bit more about that, I think, in a bit. But, when we think about the role, it really is pretty foundational. It's building that foundation for understanding and informing clinical trial development, patient recruitment for clinical trials, for really understanding, again, the natural progression of the diseases. And really, I think I've got a few bullets there on the slide, just helping to fill in the gaps.
Yeah.
Of what we don't know.
Yeah. And I think we can agree that, again, the challenges, of course, is ensuring the data quality and the standardization, like you had mentioned, but what strategies can be employed to address those challenges?
Well, there are so many challenges, and there are a few there on the slide. There were a few in the polling question, but when we look at the challenges, it does come back a lot of the time to that what's fit for regulatory purpose, and that may not be where you are in day one when you're trying to figure out, do you have 9 families or 12 families or 35 families that you can even communicate with. And so, really thinking though, I loved Kasha, your point about get started. Sometimes you have to just get started. So a lot of the things I'm going to talk about are strategies, but I don't want to in any way diminish how important it is to get started, even if it's with a spreadsheet, because look at the outcome that you had in a year from tracking with a spreadsheet. But that data quality and the ability to be able to standardize data, to control for data variability are big challenges, along with, being able to think about patient privacy, think about patient engagement over time, because it can be overwhelming even to the patients that you're asking to continue to update their experiences and share their information with you. So I think some of the strategies to address that can be things like making sure that you've got uniform protocols in place. And again, on day one, that might be 3 bullets of uniform protocols, right? And then it can grow and expand as your work in that area grows and expands. And I think thinking about things like, on day one, you may not know what drop downs to create. But after you've started to build a little bit of data, you don't want to lose those open-ended questions. But if you can start to do some simple things like adding drop downs to help standardize some of the categories or the classifications or the topics that you're trying to track, thinking about ways to train your data collectors. And when I say data collectors, yes, those can be professional data collectors at organizations that you hire or vendors that you work with. It can also be the 3 other people sitting around your kitchen table, if that's who it needs to be. But really making sure that those who are collecting and touching the data are well trained in that. And then the flip side of that is that you're educating your patients on what to expect. Why are you asking the questions that you're asking? Sometimes those questions are very personal. How do you create that safety? How do you explain to them what you're trying to do, and kind of educating them on what to expect at the beginning, but also throughout the process? Because as we know, patient registries that last for years, sometimes decades, are the ones that ultimately can often yield really, really important results. So you want to make sure folks are trained. And then I'll just add a couple other things, because we could talk about strategies all afternoon, but thinking about how do you leverage digital tools to streamline the process, to reduce error. And I think, how do you decide what to collect? And sometimes I think in the beginning, you need to collect as much as is reasonable, because you do have to figure out where that balance, where that line is, because you want to collect as much as possible, because maybe only one person had reported so far that they'd used vitamins. But then once you start to ask that question, you see that's where you find those commonalities. And so I think it's really important that you collect as much as possible, but not to the point that patients are just too overwhelmed to stay in touch.
Yeah. Really great strategies and tips we should all consider when we're thinking about collecting really good quality data. If I'm not mistaken, at this point, the Tango 2 natural history study has over 2,000 points of data that we collect now. But if you can imagine, we've been doing it for a little over 6 years now. So again, with family inputs, those outcomes that are really important to them become data points we start to consider. But we didn't start with 2,000. We got there over time. Leanne, thinking about engaging patients and collaborating with stakeholders, we know that's really crucial to natural history study success. How do you believe effective patient engagement enhances these studies?
It's all about building trust, right? You want to make sure that you build the trust, that you're, again, being very transparent in what you're asking, why you're asking, how you're going to use the information that you collect. It's all the things that the IRBs are looking for, but you need to put it in human terms, right? Like sometimes the IRB applications are a little not user-friendly perhaps for patient populations. So, the goal of them is awesome, of course, but, really thinking about how do you translate what you're looking for? And I think one of the most important things in patient engagement is staying in touch. You know, it's kind of like, one of the things I've done in my career is a lot of fundraising as well. And, I can't just reach out to my potential sponsors to support my annual gala if I don't talk to them the rest of the year. And that's also important. And I think why for both of you, when you're talking about all the other programs, that nothing is linear, that you've got lots of other programs going on, those are lots of other opportunities that you're connecting with those patients that you're connecting with through the registries. And so making sure that as your registries grow, that those families, those individuals are kind of pulled into that. Regular communication is really important. And I think too, thinking about patient engagement, making sure that they understand how important their role is. The best natural history studies are not just based on clinical data. They're not just based on research goals. They're based on human need, right? And so I think making sure that that is an ongoing communication and an ongoing priority that's lifted up, that their work is really, their contributions are really making a difference and being used in ways that they may not see day to day, but could have an overall very important long-term impact.
Yeah, very true indeed. The family stakeholder voice is so critical in shaping meaningful outcomes for our rare disease community. Finally, Leanne, can you talk about data management? Because we know it's critical to any study. How does the use of robust data management systems contribute to, effectiveness and reliability of the data that's collected in our natural history studies and patient registries?
Well, let's start by saying this doesn't have to be a huge expense. Because I think a lot of times when we start talking about data systems and data management, we also see a whole lot of dollar signs. And there are partnerships out there, there are collaboratives out there that can be very cost efficient, sometimes even free. And so first off, I think really looking at what's available to you based on the stage that you're in. You can also start with something, and then if you feel the need down the road to bring it more in-house, be able to do that. But really thinking about, the accuracy, the reliability of the data that you're collecting, to think about that ongoing monitoring. Data management, it's not just, creating that initial database, right? I mean, a patient registry is as much about looking at the long-term over time findings. And it also really helps you make more real-time refinements. You had mentioned that there were some things that you learned along the way, and like thinking now about the women's health, that was something that you didn't start 10 years ago. But being able to make that, to see those trends and be able to make those changes, having a robust data management system, it also, if you're starting a patient registry, that's a big investment. That's a big staff volunteer investment. That's a big patient investment. That can be a big financial investment. And so really thinking about, are you setting it up in a way that's going to be usable across different platforms? It's going to give you the comparisons that you're trying to get. And again, I kind of keep going back to this fit for regulatory purpose. And I wish there was and here's the checklist. And if you do these 3 things, everything's going to be fit for regulatory purpose. And it's not quite that simple. But I do think kind of keeping that as one of your end goals in mind can be very helpful.
Agreed. Thank you all so much. What we're going to do now is we're going to transition to Q&A. So I'd ask anybody that has a question just to stand up, project it really loudly. I don't know if we'll have a mic going around, but I can definitely repeat the question here in the mic.
And Deena, while we're waiting for our first question, I forgot that I had this slide. I just want to do a plug that we are working to host a hybrid public meeting on October 16th with FDA about the new rare disease hub. So we already have a couple thousand people registered. So it is hybrid, so you don't have to go to DC for the event, but it's a good way to learn from others and to be heard.
All right. Any questions? Oh, thank you. You have a mic. Lovely.
Hi, Marianne Clancy from Cure HHT. A question for you at TSC. I've known your org for a long time, and we've adopted study tracks as well. And we have we are the only organization internationally as well, but there are patient education groups. How do you think you're going to move forward globally? Do the international teams have their own registries? And how can you tie all that together? That's a challenge that we face.
Yeah, that's definitely a challenge that we're currently facing. So if you guys didn't hear, she was asking about those challenges with interfacing internationally and our associations that are at those international groups and making sure that we're collaborating with them if they already have something ongoing. So that's one of the things that we are also kind of working through. I'm working with our Director of Government and Global Relations at the TSC Alliance, collaborating really closely with her because she is kind of the TSC Alliance voice with our TSCI, our TSC international groups. And so we're kind of working currently on making sure that we're communicating what we're doing with our natural history studies and what things are going on, how we're now leveraging those data points for things that are contributing to our academic researchers and clinical trials. Our Director of Government and Global will be going to an international meeting actually next month to talk a little bit more with them about these kinds of things. So I think it's just ongoing communication, making sure that they're staying involved as we continue to grow. Our international development of the NHD is very, very new. So it hasn't started yet. It's piloting in Mexico. And so depending on how that goes, we really do plan on expanding more internationally. And so it's a very young ongoing process, but hopefully with that, these groups can see what they're able to achieve and that we continue to do things hand in hand with them. Because obviously the more data points that we can get that are diverse and representing these groups that are international, the better that the data is, but also that the more that they feel that they're represented in research. And that's important to me. So I think that it's just ongoing communication, making sure that they're involved in the process.
Okay, I have a question that came through our online audience. Does the tissue collection that you are collecting include post-mortem samples, or is this only tissue samples from living patients?
Yeah, so we do mostly collect living patients, although we are able to collect post-mortem brain tissue through a collaboration with the Maryland Brain and Tissue Bank. We are able to collect that tissue if they are able to if they're able to get in contact with the Maryland Brain and Tissue Bank once they know that death is imminent, and they're able to kind of provide all of the kits and details through that, and then they're able to give information on how the tissue will be transferred to our biobank. So that is also available. Of course, if there's any other questions, feel free to shoot me an email and I'm happy to provide more information on that collaboration.
And same for the Tango 2 natural history study as well. We collect postmortem tissue as well. And that essentially is stored at Texas Children's, Baylor College of Medicine, with whom we collaborate with to do our natural history study. Any other questions?
Hello, wonderful panel. Thank you. Abigail Spadine, genetic research, genetic counselor at Boston Children's Hospital. So I'm also involved in natural history studies, and we're really working to partner with PAGS now. But I think something we have been working on and struggling with a bit is, trying not to make this a greater burden on research participants and allowing them to not enter their data multiple times. What advice would you have for harmonizing across different research collection platforms, recognizing that not all will be IRB approved?
Do you want to take that one, Liz?
Yeah, I think in terms, I feel like I'm talking a lot. I think in terms of the burden among patients, that's one of the things that we are very hyper aware of. And of course, there's survey fatigue, already surveys that PROs that patients are filling out in clinic. And of course, if there's any other additional trials where they're completing assessments for, then they are taking surveys for that. And then on top of a natural history study that they may be participating in. So we're very hyper aware of survey fatigue and burdening them of additional things that they might need to be participating in research, which is voluntary, of course. But one of the things that we have done with the clinics that are able to extract medical records from the EMR, there's really nothing that right now with our database that we ask the patient additional fill out other than the consent form and saying that they're consenting to being contacted for research, and if there's biosample collection as well through the blood collection. So we really cover all of those study processes in terms of getting their medical records, consenting. We're able to collect biosamples at home if they're not going to a clinic through mobile phlebotomy, and we do all of that coordination. So we don't want to add that extra burden of them thinking of like, oh, what do I need to fill out for research, or when do I need to make an appointment for this? So we try and make sure that we cover all of those things, and just making sure that that's something that we communicate with all of our clinics that are participating in this. And yeah, I think just making sure that we kind of give them as minimum of things to think about as possible, because they might have someone that they're caregiving for, and we don't want to add that extra burden.
One other, if I could add,
Yeah,
one
absolutely.
other thing to just think about, and I'm sure you're already doing this, but thinking about letting them know what your ask really is. You know, is this going to take them 10 minutes, 40 minutes, 2 hours? But, you know, so that people are prepared. I think that we've seen that helps across all different types of registries and natural history studies.
I don't even go as far to say, even the wording natural history study, to some of our families, particularly internationally where language is already a barrier, what does that even mean? You know, I would love to change the title to, you know, patient experience study or something like that. You know, use words that mean something to families who they don't know what NHS is. They don't know what natural history study is. What does that mean? So that simple clarity, especially internationally, would be great.
Thank you. Any other questions? Any online?
Nope, I haven't seen any more come through online.
All right, round of applause for our panelists. Yeah. Yeah, thank you all so much for such insightful and informative discussion. The work that you all are doing is so transformative, and it's really clear, I think, from this panel that natural history studies and patient registries are pivotal to advancing the research and improving outcomes in our communities. So again, thank you so much, audience, for joining us today and being a part of this really important conversation.